this recording we'll discuss skeletal muscle contraction and relaxation. So we can break muscle contraction down into three parts: excitation, excitation-contraction coupling, and contraction. And just generally speaking, excitation phase begins when an action potential releases acetylcholine from the axon terminal and it diffuses into the synaptic cleft. Excitation-contraction coupling is the link between that stimulus, the release of that acetylcholine, and actual muscle contraction. And the contraction phase begins when calcium binds to troponin, which moves tropomyosin out of the way. So myosin can bind to actin's active sites. We call that cross-bridge cycling. That begins an actual contraction. So let's do a little bit more detail. So excitation. This neuromuscular junction hopefully is familiar. We're going to have an action potential coming from your nervous system. It's going to come on down the line until it hits the terminal. When the action potential reaches the axon terminal, there are going to be channels at the terminal. They're going to be calcium channels. This is going to allow calcium ions to diffuse into the axon terminal. When calcium diffuses into the axon terminal, it triggers the release of acetylcholine. So acetylcholine will be released into the synaptic cleft, where it wanders over to these ligand-gated channels and binds to the receptors. Now, these ligand-gated channels are going to open when the ligand binds. In this case, our ligand is acetylcholine. This opens up these sodium ligand-gated channels, because they are sodium ligand-gated channels. Sodium is going to rush into this cell, this muscle cell. This is called an end plate potential. Remember, this is the motor end plate. We are trying to turn an action potential from your nervous system. We're trying to transfer it from your nervous system into your sarcolemma. So we're trying to start an action potential at your motor end plate. We do that by generating end plate potentials, EPPs. If you have enough EPPs, multiple EPPs, you can create an action potential. If you create an action potential, you will get muscle contraction. So, one action potential comes down the terminal. We activate calcium ion channels. Calcium is going to come into the terminal, which causes acetylcholine to be released. Acetylcholine diffuses across the membrane, binds to the ligand-gated sodium channels. Ligand-gated sodium channels open up, sodium diffuses into the cell, creating an end plate potential. If we allow enough sodium to come into this cell, we will create an action potential. If we have enough EPPs, we will create an action potential. So that is your excitation phase. Now, excitation-contraction coupling. This is where we turn an action potential from your nervous system into an action potential on your sarcolemma. Those EPPs that we just talked about, bringing all of that sodium in at the motor end plate because we open ligand-gated sodium channels. This changes the membrane potential. If you change it enough, you open voltage-gated sodium channels. What happens when you open voltage-gated sodium channels? you trigger an action potential. Ooh, okay, okay. Now, once you trigger that action potential, once you open that first voltage gated sodium channel, once you trigger that action potential, it immediately gets propagated down the circle lemma. Okay, so notice our arrow. Whoop, okay. Notice our arrow also goes down our T-tubules. We are also going to trigger an action potential down our T-tubules. When the action potential reaches the T-tubules, we have more calcium channels. We have voltage-gated calcium channels here as well. These are going to open up because of the membrane change, the membrane potential change from the action potential. These calcium channels open up, and calcium is going to be released into the cytosol. Where is that calcium coming from? Your sarcoplasmic reticulum. Okay, so don't forget about your good old SR. Okay. The terminal cisterna of the SR, remember, are packed full of calcium. That calcium is going to go through these calcium channels, and it's just going to go into the cytosol. Notice, okay, do not make this mistake. Everybody pay attention. You ready? Okay. This calcium going from the sarcoplasmic reticulum, it goes through these little channels. It does not go into the T-tubule. Notice there's no little green dots in the T-tubule. Okay. Calcium moves from the SR. It goes through the calcium channels and it comes into the cytosol. Okay. They should have put a bunch of green dots out here as well to really hit that point home. Okay. They're here. They're not in the T-tubule. Okay. So now that calcium has been released into the cytosol of your muscle fiber, what happens? Okay. Calcium, oh, here's a better picture. Okay. So remember, all these calciums were in the sarcoplasmic reticulum. We're going to go through the calcium channel. Okay, here's the channel open. All the little calciums are in the cytosol. What are they going to do? They are going to bind to troponin. Okay. Once calcium binds to troponin, tropomyosin gets pulled out of the way, which opens, okay, or exposes the actin active sites. When the actin active sites are exposed, guess what can happen? Myosin heads can finally bind to the actin active sites. Okay? Which is our last step, contraction. Okay, so for contraction, the myosin heads become energized. Okay, good old ATP. We're going to hydrolyze ATP. The energy from ATP gets transferred from ATP to the little myosin neck. Okay, charging them up. Because we have already moved tropomyosin out of the way and those active sites are now available, we have energized our myosin heads and necks. Myosin attaches. We call this cross bridge formation. Okay. And then we pull. We do our power stroke. Okay. Notice that we have hydrolyzed ATP and it becomes ADP. Okay, the energy has transferred from that molecule into the myosin itself, which gives it the energy to pull. Okay, we're going to pull once we release the ADP. We don't really need him anymore. Okay, we're going to pull on the actin. We're going to pull everybody towards the M line. Okay. If we want this myosin head to let go and reach farther down and pull again, we're going to need more ATP. And we can just do this over and over. Remember, it's cross-bridge cycling. It's a cycle. Do it over and over and over. Okay? So, one more time. Okay? We have already moved troponin and tropomyosin. Everybody's out of the way. Actin active sites are ready to go. So, we energize our myosin heads by hydrolyzing ATP. It becomes ADP, and that energy moves into the myosin. Myosin reaches up and grabs on to the actin active site. Okay. And then we pull. We've made that cross bridge. We're going to pull. We're going to do our power stroke. We have to let go of ADP in order to pull. Okay. Now that we have moved this actin towards the M line, if we would like to let go, okay, say we want to let go and we want this myosin head to reach out to this next one, okay, we need more ATP. Okay, another ATP will bind, we can hydrolyze it, and we could instead maybe pull here and do it again. Okay, so that is muscle contraction. It seems like 25 million steps. I know. I wish I could tell you only needed to know 10 million of those steps, but you need to know all 25 million. Okay? Make sure you go back step by step by step. Now, in order to do muscle relaxation, okay, we need a couple things to happen. Okay? First of all, your motor neuron has to stop sending more action potentials. Okay? If your brain keeps sending signals, okay, we're going to keep releasing acetylcholine. It's going to keep binding to the motor end plate. You're going to have more EPPs. You're going to keep making action potentials. Yada, yada. You're going to keep on doing the same thing. Okay? So we're going to have to stop sending action potentials down the motor neuron. Okay? You're also going to have to stop releasing more acetylcholine. Okay? Get rid of everything that's in the cleft. Put everything back where it used to be. And last but not least, calcium has to get pumped back into the SR. Okay? You're going to have to actively do this. You're going to have to go against the concentration gradient. Okay? We're going to have to use some ATP here. But if calcium is still in the cytosol, it will still bind to troponin. It'll still expose the actin active sites. You'll still have cross-bridge formations, yada, yada. Okay? So, picture. First thing, we'll have to stop sending an action potential down the terminal. Okay. We'll have to stop releasing acetylcholine. Okay. Any acetylcholine that is already in the cleft, we're going to have to get rid of that. We have an enzyme called acetylcholinesterase. Okay. That acetylcholinesterase will degrade any acetylcholine in the cleft. No more acetylcholine means no more activation of ligand-gated channels, which means no more end-plate potentials. Repolarization can finally start. Okay? Remember, in order to achieve repolarization, okay, we will have changed the membrane potential. We will have closed the sodium channels. We're going to need to open potassium channels. Okay? And again, don't forget about the sodium-potassium pumps. I don't know why they never get included in the drawings. It's kind of hurtful, hurts their feelings. But the sodium potassium pump helps restore resting membrane potential as well. Okay. Since we are returning to resting membrane potential on the sarcolemma, that includes the T-tubules. Okay. So that closes the calcium channels. No more calcium is being released into the cytosol. No more calcium binding to troponin. The tropomyosin covers the actin active sites again. Myosin can no longer form those cross bridges. We can't do any more power strokes. Okay. Since we can't do any of that anymore, the myofilaments return to their original length. We achieve muscle relaxation. Okay. Now, when you die, everybody's heard of rigor mortis. Okay, your body gets stiff when you die because you are in a constant state of contraction. This happens anywhere between about 3 and 4 hours after your death. Okay, begins. Why? What's going on? What's happening? As the pumps that were pumping calcium, okay, back into the SR. Well, they don't have any more ATP because you're dead. Okay, so we can't pump any more calcium back into the SR, which means the calcium just sits there. I just told you if the calcium is still there, they're still going to bind to troponin. You will still have cross-bridge formation and power strokes. You'll still have muscle contraction. We also alluded to the idea that if you wanted to move your myosins again, you would need more ATP. Well, you don't have any more ATP. Okay, so the myosin heads cannot detach from the actin, so they're just stuck. Okay, they're stuck in a contraction period, and they will stay that way until the myofilaments themselves start to degenerate or degrade. Basically, your body starts to break down itself. And this happens anywhere between about 48 and 72 hours. Okay, so you start to get stiff 3 to 4 hours after death. Okay, you stay stiff for about 2 or 3 days, and then you just go floppy. You don't really care, you're dead. It's okay. All right, putting it all together. Okay, putting it all together. Excitation, we took an action potential from the nervous system. Okay, the calcium that entered the axon terminal caused acetylcholine release. Acetylcholine binds to ligand-gated sodium channels, which creates end-plate potentials. End-plate potentials change the voltage enough to open voltage-gated sodium channels, which starts an action potential. The action potential moves down the T-tubules, and we open voltage-gated calcium channels. Calcium leaves the SR, goes into the cytosol where it binds to troponin. Troponin moves tropomyosin out of the way to expose the actin active sites. Okay. Myosin heads will hydrolyze ATP to energize themselves, will bind to the actin active sites, cross bridge formation, and we do our power stroke, we pull. Okay. Remember, at some point we had to let go of that ADP as well. If we would like to do this again, we need to bind a new ATP. We'll hydrolyze them. We'll just keep doing this over and over and over. If we want to do relaxation, then we have to undo everything. We stop sending our action potentials. We get rid of our acetylcholine. No more end plate potentials. We get to repolarize. Our sodium channels have closed up. We open our potassium channels instead. The change in membrane potential closes these calcium channels. The calcium gets pumped back into the SR for storage for later. The calcium is now gone, so troponin, okay, kind of undoes everything. Tropomyosin covers the actin active sites again. Myosin can't bind. Everything gets to relax.